You know about the nomenclature of the carboxylic acid derivatives and their properties. Let's talk about their reactions and their synthesis. Okay, we'll take it all together. So again, go get your cup of coffee. So on this slide, what you can see is all the derivatives of the carboxylic acids. So you won't see the acid here because we've already done that in a separate PowerPoint. So in this one, you will see the acid chloride, anhydrides, esters, amides, and nitriles. Okay, so these are all the derivatives we will talk about. What this also shows you is the relationship to the acid to the derivative. Okay, so for example, if you see here for the acid, okay, and the acid chloride derivative, if you take the acid and treat it with HCl or SOCl2, for example, thionyl chloride, then if you remove water, you will get the acid chloride. If you take an acid, two acid molecules, and you remove water, you get an acid anhydride. If you take an acid and an alcohol, remove water, you get an ester. If you take an acid and an amine, remove water, you get an amide. If you take an enol of an amide, okay, which amide is over here, and you dehydrate that, again, you remove water, you get a nitrile. So what this little diagram tells you, all these reactions tell you, is that there is a direct relationship between the acid and all of its derivatives. So what happens is that the majority of the reactions that we will talk about in this chapter or in this little topic is how do you convert from one derivative to another derivative and so on, because as you can see that almost all of these derivatives can be made from dehydrating one or the other combination of acid, okay, with something else. So let's look at the kind of reactions that you will get for all the derivatives. Okay, there are two kinds of reactions that you will get. One is the nucleophilic addition and the other one is nucleophilic substitution. Nucleophilic addition is the same thing as you did for aldehydes and ketones, where the nucleophile attacked the carbon, electrons moved on the oxygen, and the nucleophile then will attach to the carbon over here, and the oxygen here, which was the carbonyl oxygen, will form an alcohol. So this would be an addition product, and again the nucleophile is still there. Okay, and so this is a nucleophilic addition reaction. You will almost end up forming alcohols for where the carbonyl oxygen was. Okay, in the substitution reaction, it's like any other substitution, like SN2 substitutions, where you have a nucleophile attacking and then you have to have a leaving group. Okay, and so your reactivity of the derivative will depend on what kind of a leaving group it is. Okay, but anyhow, what happens is that the carbonyl oxygen is retained. Okay, in this case, so you have the leaving group that goes away, nucleophile that replaces the leaving group, okay, and the carbonyl states. So which means that your reaction here now is going to depend on the strength of the nucleophile and the ability of the leaving group to leave, okay, which means this would be a weak base. The weakest base possible is that from chloride, okay, because chlorides are weak conjugate bases, so which makes acyl chlorides really very reactive because this is a good leaving group. Acid anhydrides are next because the leaving group here is the carboxylate group, okay, which is this one. So that would be the next one in line. Esters would be the next one in line, as in OR minus, which are alkoxide. They're not the best leaving group, but they can be made into a good leaving group depending on the kind of nucleophile you're using. If it's a good strong nucleophile, then yes, this will go away. Okay, and then the least of all are the amides. Okay, and the amides are really very unreactive because NH2 minus, the amide group, is a terrible leaving group. Why? Because it's such a strong base. Okay. The acids kind of fall right here. Acids meaning carboxylic acids, which would be COOH. In COOH, the OH, hydroxide, is the leaving group. So again, hydroxides are not a very good leaving group. So hydroxides actually fall right here right along with the amides. Okay, so acyl chlorides most reactive, amides least reactive, and it's shown nicely in the chart on the next slide. Here is your reactivity, and I included the acid over here, although we will not be talking about the acids too much in this little presentation, but all the other derivatives, yes, we will talk about. Okay, so but the acids are here just for your reference. So acyl chlorides best, amides are then the least reactive. The interesting thing about acid derivatives is that there is a whole lot of interconversion going on between them, okay, which means you can take the most reactive acid derivative, which is the chloride, and convert it into anything after that. So anhydride, again, you can take that and convert it to anything after that. The only thing you cannot do is convert anything right back to what it was before. Okay, so for example, you can't take an anhydride and make it an acyl chloride, all right? Or you can't take an ester and you cannot make it an acyl chloride, or you cannot make it an anhydride. However, from anything that's on the top, we'll give anything at the bottom. The only thing that goes way back is carboxylic acid, and on treatment with thionyl chloride it will give you the acyl chloride, but then again that is a very specific reaction, okay, for carboxylic acids. So all of the other derivatives, this is kind of how they fall through in the chart of reactivity, okay, with the amides right here at the bottom, and carboxylates, no, we don't do any reactions with carboxylates. So let's start with the acyl chlorides. The only way to make acyl chlorides is actually using a chloride nucleophile. Okay, and the most potent chloride nucleophile comes from these three reagents: thionyl chloride, or phosphorus trichloride, or phosphorus pentachloride. So you can use any three of these reagents to convert an acid chloride into an acyl chloride. Okay, and so once you have the acyl chloride made, then you can do all sorts of reactions on it. But this is the only reaction, or this is the only way to make acyl chlorides. We did this reaction back in acids also. So here are the reactions that you can have of acyl chlorides. And again, remember, acyl chlorides are the most reactive of all the derivatives. So they will form anything that is underneath it, okay, which means are less reactive than that. So the first and the foremost thing you can do for any kind of acid derivative is hydrolysis. Okay, remember that slide that we just saw previously, is that everything is coming from dehydration of of an acid. So which means you can add water and go right back. All right, and so therefore we're going to do this reaction on almost all the derivatives, okay? And what I'm going to do with you here with these reactions is, first we will talk about all the interconversions between acid derivatives, and then we will talk about some specific reactions. So here we have the acyl chloride, which on hydrolysis, whether you take acidic or basic hydrolysis, it doesn't matter, and you will end up getting an acid. So here is in a basic hydrolysis, and again, if you treat this with water or acid, it will give you back your acid. So hydrolysis will give you back the acid. The rest of the reactions that are given over here are for all the other derivatives that we work on. So for example, if you take the acyl chloride and treat that with a carboxylate, any kind of a salt, okay, like sodium acetate, sodium formate, whatever it is, then you will get an anhydride. Okay, so remember, after acyl chlorides is anhydride. After anhydrides is the ester. So which means you can take an acyl chloride, treat that with an alcohol, and you get an ester. Treat the acyl chloride with ammonia or any other derivative of ammonia, which could be an amine, and you will get amides. Okay, so these are just the two down here are just substituted amides, but otherwise you would get amides in all of these three cases. Okay, so which means hydrolysis, formation of anhydride, ester, and amide. So you form all the three derivatives over here. For this one, you can look up the mechanism in the textbooks, but I don't cover it so much. Anhydrides, then that's the next one on the list. So now what happens is that you can make anhydrides from acid chlorides, okay, because we just covered that. So you take an acid chloride and a carboxylic acid and you will get an anhydride, okay, or you can take a carboxylate, which is a good nucleophile, fairly okay nucleophile, along with an acid chloride, and then you can also get an anhydride. The other way you can also make anhydrides is that you can take a diacid, or you can also take an acid like this, aliphatic acid, and heat it up a little bit. And once you heat it, then you will have dehydration, so water will go away, and then you form an anhydride. So this is a very good way of making cyclic anhydrides. Okay, and by the way, if you're trying to make anhydrides using this method, then you can make only symmetrical anhydrides. With these two methods above, which is acid chlorides with carboxylic and acids, with these ones, you can make different kinds of anhydrides, which means that you can have two different fragments on either side of the oxygen. But in these cases, with the heating part, then you can only have one kind. Okay, so it's a symmetrical anhydride. So if you want to make this, this is the easier way to make it. Now for the reactions of anhydrides, again, hydrolysis. When you do hydrolysis on anhydrides, you will get two fragments now. Don't get confused by the two fragments, okay? In the acyl chlorides, the chloride was the leaving group. In anhydrides, this whole green part here, the carboxylate, that is the leaving group, okay? So that's the leaving group that you form. In some cases, if it's an acidic medium, it will be protonated. If it's a basic medium, it will not be protonated. So you will see it as an ion, and that's okay, all right? Don't worry about it. It's just that it's in ions, but otherwise you can protonate it easily, okay? So here's the first reaction that you will see for anhydrides, which is hydrolysis. The one that is missing over here is, of course, synthesis of anhydrides, remember, from the acyl chlorides, because now that has gone into the synthesis of anhydrides. But anyhow, you can take anhydrides and then treat that with alcohol. And if you treat that with alcohol, for one fragment, you will get an ester. The other fragment will go as a leaving group and form an acid. Okay, so this will be in acidic medium. So you get an ester as well as an acid. With ammonia, you will get an amide as well as the carboxylate, and this will be a salt, okay, over here. And the same thing that you will see in these two cases where you're dealing with substituted amines. So the only reaction you don't see over here is the synthesis of acyl chlorides, and you don't see that because acyl chlorides are more reactive than anhydrides. Esters now. For the synthesis of esters, okay, there are few syntheses now, because as you're going forward into the derivatives, now you can make esters from acyl chlorides, you can make them from acid anhydrides, and then you can also make them from acids. Okay, that is also one of the ways to make them, and we talked about this one in the previous PowerPoint, in the previous lecture, where we had acid reactions. Okay, so if you want to review that, you can look at it there. We didn't do the mechanism, we're going to do it now. Okay, so so there are three syntheses, okay, that we will talk about here. So the first one is the acid catalyzed reaction between alcohols and esters. Okay, this is called Fischer esterification, and Fischer esterification is a very neat way of making esters. All you need to do is get your appropriate alcohol and acid, and then heat it up with a little bit of acid, like sulfuric acid, for example, and then you end up forming an ester. Okay, and so it's a nice little synthesis. Sometimes you may need to heat it for an hour or 2 hours, but that's about it. So the OH over here, okay, that will be the leaving group, and the alcohol here will do the attacking. Okay, and we'll just talk about the mechanism over in the next slide. But these are some examples, okay, of how you form the esters. I want you to note one thing over here, okay, because this is a highly studied mechanism, because it's one of those mechanisms that occurs a lot in our body. I want you to notice the pinks and the blues, okay, and the blue part here is coming from the acid, which is of course the carbonyl part. That fragment is coming from the acid, and then there's the other part over here that is coming from the alcohol. Now the thing about what I want to point out here is about this oxygen, this black oxygen, or the oxygen which is attached to the hydrogen. This OH completely is a leaving group. The OH from the alcohol is not the leaving group, okay? The OH of the acetic acid is the leaving group, all right? So this is something that you will see in the mechanism, okay? Is that when you get water, okay, from here, where is the oxygen coming from? Is the oxygen coming from the alcohol, or is the oxygen coming from the acid? That is very critical to know the pathways, and especially when you're studying biochemistry, then you really need to know the pathways of how these things work. So that's why we do mechanisms, all right? And you will see later on when we do the mechanism that the water is coming actually from the OH of the acid. Okay, all right. So let's look in detail with the mechanism of esterification. So here is the basic equation, well, acidic, but fundamental equation, where you have the acid and you have the alcohol giving you the ester. Now, one of the most common ways of following reactions in biological systems or even otherwise for determining mechanism is to radio label certain atoms so you can follow it through. So what I was talking about before as to where the oxygen is coming from for the water, whether it's coming from the acid or whether it's coming from the alcohol, well, it was determined by radio labeling the oxygen of the alcohol, and then you follow it through. So it turns out that the radio labeled oxygen here in the alcohol was found in the ester. So if it's found in the ester, then you can predict the mechanism a little bit more and say, okay, well, then probably the water is coming from the OH of the acid and H of the alcohol. Okay, so let's look at the mechanism now. You start the same way as you would for an aldehyde or a ketone, where you have a carbonyl getting protonated from the acid, because this mechanism is occurring in acid. By the way, this mechanism can occur also in basic medium, but we are going to do the acidic one. So here it is getting protonated. So the oxygen now is protonated, which makes this carbon a little bit more electropositive, easier for nucleophilic attack, which is what happens here with the alcohol. And so the alcohol will get attached here, electrons move to the oxygen. So now this one is relieved of the oxonium ion. And now what happens in this step here, okay, is where you have the methanol that attacked. That hydrogen is still attached. Okay, this different colored arrow that you see over here is the one that I have drawn because it was not in the original mechanism. Is this oxygen will pick up this proton, which then this different colored arrow, the proton would leave its electron on the oxygen. So this is how you get this species over here. In which case now this one is deprotonated. However, this OH now is protonated. So now you can see that where the water is going to be coming out of. Okay, the water is not coming from this oxygen, but the water is coming from this oxygen, which is what we said over here. Okay, and so this is how it gets protonated. So then we have to get our carbonyl back, all right? And so this oxygen again, the carbonyl oxygen, all right, will give its electrons back to the carbon. And then here the electrons are transferred to the oxygen. This water goes away. And then this oxonium ion now has to be relieved. So the water will come and pick up a proton. And that's how you get the ester. Okay, this is one of those mechanisms that you really have to know. Back and forth. Okay, so forward as well as backward, because if you see all the arrows in this mechanism, they're all reversible in equilibrium. So you can push the equilibrium forward if you add acid, and you can make it go backward if you add water. Okay, so just like any other equilibrium you have learned in Gen Chem 2, forward with acid, and then to push it backward you would add more of the product, which is water. Okay, so you should write this mechanism on your own with water also. Okay, and so in case of hydrolysis, which would be going backward to give you the acid and the alcohol, you would start from protonating this oxygen and then follow each of the steps back, okay, to fragment right here. When you do this attack here for the reverse reaction, this is where water will be attacking also. Okay. So the other synthesis of esters will be from acyl chlorides, as you've already seen before. The pyridine here is for catalytic purposes, okay? And so the acyl chloride will react completely with alcohol, no problems there. But in case you want to facilitate that a little bit better, then you can add a little bit of base, okay, which pyridine is a slightly weak base. You can also make esters from anhydrides, because remember, anhydrides are more reactive than esters, so you can make it from there also. And so these are a couple reactions showing you how this works. Now this anhydride here is the acetic anhydride, which is an aliphatic anhydride. This anhydride here is a cyclic anhydride, and it's a good thing that you have these two, so you can see how when you have an aliphatic anhydride, you have a nice leaving group here for the acid. But when you have a cyclic anhydride like this, you will not have a leaving group, because this carbon is still attached to this carbon, which is part of the original compound, the phthalic acid. Okay, and so the leaving group is sort of over here, but not really. It's not getting fragmented into two like this one. Okay, in this case, you would get two fragments. One is going to be a typical leaving group, but here you don't see the second fragment because only this bond has broken. Okay, excuse me, this bond has broken. So that's the only thing you see over here. This fragment is still attached to this carbon. Okay, so there is no second fragment. And then, of course, we have lactones. Lactones are cyclic esters, and cyclic esters can be formed if you have an acid and an alcohol in the same compound. Okay, so depending on how long your chain is, that is how long of an ester you will get. Now remember that oxygen is part of the ring. Okay, this oxygen is part of the ring, and this oxygen is coming from the alcohol. So again, you start the mechanism of esterification just like normally you would for Fischer synthesis, is protonation of the carbonyl. Once it's protonated, then this alcohol will attack this carbon, and you will get this kind of a fragment. This looks like an acetal, but it's not really an acetal, okay? And this is kind of an intermediate here. So then you have this protonated alcohol. Now remember, one of these has to go away, all right? And so you'll have water leaving the way. This OH has to go, okay? This OH is, well, you can think of it as this one, all right? And so this OH will get protonated using this hydrogen. And once it's protonated, then you can have removal of water, and then you can restore your carbonyl carbon with the oxygen here, okay? And so you end up getting a lactone. So this is the forward reaction, and then you can do the hydrolysis also of lactones, and I just wrote it over here. So you have it all on the same slide rather than on two different ones, okay? So you can see how it opens up. And again, you can use acidic or basic medium, okay, for hydrolysis. But when you open up lactones, again, you will get one long chain because the alcohol and acid are both part of the same chain. Okay, so this is opening, this is reverse of synthesis. You'll have to do both mechanisms. The reactions of esters in general is hydrolysis first, okay, and as we said before also, every reaction, every derivative that we have will undergo hydrolysis. So the hydrolysis of lactones we just talked about, and this is hydrolysis of esters. So once you do hydrolysis, acidic hydrolysis, for example, then you will have an acid, and of course you'll have the alcohol as the leaving group. The OET is going to leave as the alcohol. You can also do transesterification. Okay, so this is a very common thing that happens in the body also, and in some cases in the lab we do this because certain groups are good leaving groups or easy to get rid of than others. So for example, in this case where you have this ester, which is a methyl ester, and you want to change it to a butyl ester, you can easily do that, okay, by taking an alcohol of your choice, whatever you want to convert it to, and using acidic medium again, and you can get transesterification. You should try and write the mechanism, okay, for this reaction instead of water or any other alcohol, you would be using the other alcohol here, okay, and then this will be your leaving group here. In case of acids, your OH was the leaving group as water. Now OCH3 will leave as alcohol as methanol, okay, over here. So you get two fragments. So try the mechanism of this one as well. The third reaction of esters, which is kind of unique to esters, you will not find that for any other functional group, is saponification. And saponification is when you take an ester and treat it with a strong base. And this is like an acid-base reaction if you think about it. Carboxylic acids react with bases, okay? But esters also tend to react with strong bases, and they end up giving carboxylate salts. And these salts are just like any other salt, okay? But this is a very common way of making soaps, actually. You would not make carboxylate salts using this method, okay? You can use acids for that. Then we get to the next functional group and one of the last ones, and that is the amides. Okay, and amides can now be synthesized from all the functional groups above, which means the chlorides, the anhydrides, the esters, okay, all of them, and also from acids. Okay, so from acyl chlorides here, this is how you would make the amide. You would take any derivative of ammonia, okay, NH3, but here the R groups are any other different alkyl groups. They could be the same, they could be different, whatever it is. But using acyl chlorides, this is how you would go about the reaction. Remember, chlorine here is going to be the leaving group. This is a classic SN2 substitution, okay, not SN2, but this is a substitution reaction. And then you can also make it from anhydrides. Now in anhydrides, there is no leaving group. There is a leaving group, but not in this case, okay? In anhydrides, the leaving group would be the entire acid, but in this case, because this is a cyclic anhydride, your leaving group is still attached to the benzene ring, okay, over here. So, but then here is your amide, okay, that you just formed. And here is the leaving group, which is still attached over here to the benzene ring. But if it was an aliphatic anhydride, then you would see this as a leaving group. And then from carboxylic acid, you can form amides also. You definitely need some sort of a coupling reagent or some catalyst in order to do this reaction. Otherwise, this is not a very good reaction. Okay, as we said before, also carboxylic acids are not very reactive. Okay, so they can work. People like to work with the acids because they're readily available, but they're not very reactive. But you can do this reaction using some sort of coupling reagents, and you don't have to worry about what reagents they are. Just know that you can make them, okay, with some kind of a reagent. But anyhow, you can have amines here, ammonia, whatever you want. So these are all the synthesis for amides. The reactions of amides is very straightforward, okay, which is just one, that is hydrolysis. Okay, pretty much that's the only thing it will do as far as converting it to another derivative is concerned. So on hydrolysis, you end up getting an acid, and of course you get the ammonia back. Now, because this is an acidic medium, the ammonia here is ammonium. It is protonative. When it's neutral, then you will get ammonia. So go through the mechanism of the hydrolysis. I don't want to go through this. It will make the presentation too long. There are some other reactions we need to go through. And don't forget, with amides, you also have lactams. Okay, I wrote it on the previous slide that you should try and make some lactams also. With lactams, you need an alcohol group, an acid group and an amine group in the same compound. Okay, that's how you get a lactam, which is a cyclic amide. So here it is again, our interconversion chart, okay, where you have the acyl chlorides as the most reactive and amides as the least reactive. So we just finished with amides, and so here are all the reactions for you again, okay. Now the nitriles. We didn't really talk about nitriles yet, okay, because nitriles don't have the oxygen in them, so I kind of kept them at the end. They also don't have the traditional leaving group as we talk about in the other derivatives. So I again, that's the reason I left them at the end. So how do you make nitriles? You make nitriles from alkyl halides just using SN2 reactions, and you've done some of these reactions before, okay, so this is not something new for you. So for example, if you have an alkyl halide like this, then you would go ahead and use sodium cyanide or any kind of a cyanide compound and do an SN2 reaction, get a nitrile. We also made nitriles from aldehydes and ketones. So we took a carbonyl compound and we treated it with cyanide, okay, and that's how we get the nitrile, okay. And then lastly, you can also make nitriles from dehydration of amides. That was a reaction I showed you on almost like on the first or the second slide. Okay, so here you can take an amide, heat it up with something like thionyl chloride or something else, maybe sometimes even a phosphorus reagent, and then you can get a nitrile as well. Okay, don't worry about the mechanism, just know that you can form a nitrile, okay, from dehydration of amide. So those are the three syntheses of nitriles. The reactions of nitriles as an acid derivative is only one, okay, which means the only thing you can do on nitriles as an acid derivative is hydrolysis. And again, we did this back in aldehydes and ketones. So you take a nitrile, you treat that with dilute acid, and you will end up getting an acid, all right? And then the NH2 comes off as ammonium ion, okay? So that is hydrolysis. And this covers pretty much all of the reactions that you have for acid derivatives. Now we have some other reactions where all of these derivatives are not really converting from one to the other, but it doesn't mean they don't undergo any other reaction, okay? So some of these carboxylic acid derivatives will also react with Grignard reagents, some other organometallics, and then we'll talk about reduction also, okay? And so the first reaction here let's talk about is carboxylic acid derivative with Grignard reagent. Grignard reagent is a fairly good nucleophile, okay? And so because this is a fairly good nucleophile, it will take whatever acid derivative you have all the way to the alcohol, which means that you need 2 moles of the Grignard reagent, okay? One here, one here. The intermediate that you form is a ketone, all right? And the mechanism is on the next slide. But you don't really isolate the ketone at all. Okay, you use the acyl chloride in such a way, and the Grignard reagent in such a way that you use them in 1 to 2 mole ratio. So you'll end up getting alcohol. You try not to do this reaction in order to make a ketone. Okay, you would not use Grignard reagent for that. But anyhow, this is what happens when you have acyl chlorides. For esters also, you will end up getting a tertiary alcohol. Okay, so if you take any ester, treat that with the Grignard reagent, 2 moles of this, and you end up getting a tertiary alcohol. We do not use acids for this reaction, and I want you to think about why we do not use acids for Grignard reagents. So here is the mechanism of the reaction, and you were supposed to write this on your own. Hopefully you did. So here is the Grignard molecule coming in over here, the nucleophile attacking the carbon. And we've done this mechanism before also, okay? And then the oxygen coming down with the electrons here. The OCH3 can leave easily now because this is a weaker base than an alkyl group. So this leaves and you get a ketone. The ketone again then get attacked by the Grignard reagent, just like a normal reaction that we've done before in the synthesis of alcohols and also in the reaction of aldehydes and ketones. So now this alkyl group will attack over here, and that is how you get the alcohol. Okay, and that's how you get 2 moles of Grignard reagent onto your tertiary alcohol. And then we have the copper reagents. Okay, now with copper reagents, things go a little bit slower. Okay, I mentioned to you before also that when you treat anything with copper reagents, you have a little bit more control over the reaction because now the R group here, the alkyl group, is not so nucleophilic as it would be if it was a lithium reagent or a Grignard reagent. Okay, in a copper lithium reagent, the R group, the alkyl group, is a little bit less nucleophilic. So if it is a little bit less nucleophilic, you can control the reaction a lot more. So now the acyl chlorides will give ketones. So this is a good way of making ketones if you wanted to from acids. Okay, and so this is the reaction. Don't worry about the mechanism so much. Just know that acyl chlorides will give you ketones, okay, with the copper reagents. And then we have the reduction. Okay, we've talked about reduction before also because we have reduced carboxylic acids before as well. All carboxylic acid derivatives will typically give you primary alcohols because carboxylic acids are terminal groups. Okay, so you can reduce acyl chlorides, and acyl chlorides will give you alcohols. Amides will give you amines. Nitriles will also give you amines. Okay, so here this is what's going on with these 3, and I put the other 2 reactions with esters on the next one because the mechanism is there too. Okay, but this is a reduction of acyl chlorides and amides and nitriles. The esters are a little bit different. You have to be careful here, okay, because you can do 2 kind of reductions with esters. One with a strong reducing agent and one with a weak reducing agent. So if you use a strong one, and by the way, you can do that even with acyl chlorides. If you use a strong reducing agent, it will take you all the way to the alcohol. If you use a weak reducing agent, it will take you to the aldehyde, say like diba or dibal, okay, whichever one you want to call it. Okay, but you don't want to use lithium aluminum hydride, okay, for that, or LAH for that. You want to use a weak one that will take you to the aldehyde. And you can use an ester. You can also use acyl chlorides to do this kind of reduction. Here's the mechanism. And in the mechanism, the hydride is the nucleophile. Okay, hydride is a very strong nucleophile. All right, and so it easily does this reaction, and then OR, which is the alkoxide, is going to be a leaving group, which when it gets protonated becomes an alcohol. Okay, so this is how the reaction works. I need you to remember this main thing. If you don't remember the reaction mechanism, it's fine, although it's very easy. Just like the Grignard reactions, this is exactly the same mechanism. So here are all your reactions that you've done so far in this chapter. So we have the acyl chlorides, okay, and the acyl chlorides, these are all the reactions that we talked about. Here are the esters. We talked about all of these reactions. The last one here is the saponification. Here is the anhydrides. These are All the reactions, the amides and the nitriles, we have very few reactions of those, okay? But all the others are covered here. Acids are not here because acids was a separate chapter, okay? So go through those, keep this chart handy because it's really interesting to see how you can do one reaction with one type of derivative but not with the other one, okay? All right. Practice this on your own, which is interconversion. So you take an acid, convert it to acyl chloride, and then convert it to different kinds of groups, okay, over here. So try to do this on your own, and we can talk about it in class. You can see the key concepts are only two because really, yes, all you're doing is remembering the synthesis and reactions of all the derivatives, everything, yes. The mechanism that I ask for most is the esterification. In esterification, it's everything. So I ask for esterification, hydrolysis, trans-esterification, lactones. I can give you anything about esterification. But all the reactions also, of all the derivatives, and this also includes acids, okay? Don't forget acids.